Hello and welcome to my channel Omni Bioentrance, and today we will be discussing the CSIR Net Life Science question paper. This is part C questions. This was conducted in the year July 2025. So coming to the first question, they have given two graphs here and they have asked questions regarding and they have given statements regarding these graphs. This is a flow cytometer data and you have to see for all the correct statements. So if you see figure A, you can see there is overlap between the peaks. So this peak I am taking as A, this peak I am taking as B, and this peak I am taking as C. Similarly here also this is A, this is B, and this is C peak. So here if you see there is overlap between the peaks, whereas these peaks they are separated. Now here you see the peak width is also very large when compared to these peaks. Okay, here it is very large. So this overlapping and this broad width, it shows that there is variation within the group. That is, there is variation in, this is A group. So this peak is A group, this peak is B group, and this peak is C group. So there is more variation within the group. Okay. So if you see there is more variation within the group than between groups. That is, between A and B or A and C or B and C, the variation is less when compared to variation within A alone. Okay. But here if you see, the variation within each group is less. But between groups there is more variation. Okay, now read the statement. So figure A represents three normal distribution. Yes, since it is a bell-shaped curve, it is a normal distribution curve with more variation within the group. Yes, more variation you can see within A itself, or you can see more variation within B group, or you can see more variation within C group than between group. Between A and B there is less variation. Okay, because overlap is there between the groups. So there is less variation within groups than between groups. So A statement is correct. Then figure B represents three normal distribution with more variation between groups than within groups. I just explained that also. So their peaks are separate. So within group variation is less when compared to variation between groups. So between A and B or A and C, variation is more. Okay, but within A the variation is less. Then the ANOVA F test is likely to be statistically significant when applied to figure B but not to figure A. So ANOVA test, it is used to determine if there is any significant difference between variables or between the means within a group. Now it will show significant variation between the groups if the variation between groups is larger than within the group. So variation between groups should be larger. So variation between groups, the between groups means between A and B, A and C, like that. So it is larger in the case of B graph than in A graph. So ANOVA F test will show that the statistical significance for figure B than to figure A. So C is also correct. So your answer is 4, that is A, B and C. Then before coming to next one, I will tell you regarding the CRISPR-Cas9. They have asked questions regarding CRISPR-Cas9. So what is CRISPR-Cas9? So CRISPR-Cas9, it uses guide RNA as well as there is the Cas9 protein which will bring about a double-stranded cut in the DNA. So scientists they can bring about gene editing by using this CRISPR-Cas9 technology. So for that the scientist will design a guide RNA sequence which is complementary to your target DNA. So this is the target DNA, this is the PAM site, and this is the guide RNA sequence which is complementary to this particular target DNA. Then this gRNA or the guide RNA they will synthesize in the lab, insert it into a plasmid. The plasmid will go and infect the cell which is having your target DNA which has to be edited. So the target DNA, it should have a PAM sequence next or downstream of the target DNA. It is to this PAM sequence that the Cas9 will be able to bind. So this guide RNA Cas9 complex is introduced into the target cell with the help of plasmid or any other vector. So this Cas9, it will recognize the PAM sequence on the downstream of the target DNA. Then it will cause unwinding of the target DNA, and this guide RNA will scan the target DNA sequence, and if it is complementary, that is full complementation should be there. That is the target sequence on guide RNA should be 100% complement with that of the target DNA. So in that case, the guide RNA and target DNA, they will form complementary base pairing. The Cas9 will come to know that the guide RNA has formed complementary base pairing, and it will bring about double-stranded cut. So this is the target DNA which is cut by Cas9. So it is a double-stranded break. Now the cell can use its own repair machinery for repairing the double-stranded break, and you can bring about modification there according to your type of editing you want. So it can be of two types, that is the repair pathway can be of two types, that is homology directed repair and non-homologous end joining repair. Now if it is homology directed repair, this is the target DNA. So into that you will add a donor template. If you want homology directed repair, then you will add a donor template. The donor template will have a sequence of your choice which you want to introduce into the target DNA. Okay, so that is how you use the cell's homology directed repair machinery for introducing your gene of interest into the target DNA. Okay, so this desired insert, it will bind to this target DNA sequence because the desired insert will have homology regions here. So this region will be will form homology pairing with the donor template. Your desired insert, whatever you want, it will be introduced into this target DNA. So that is modified target DNA that you get by homology directed repair. Then the next is the non-homology end joining. So here it is a random process. So there are certain enzymes which bring about a direct joining of this double-stranded breaks. So when you use non-homologous end joining, they can bring about this insertion or deletion mutations in the DNA. So this is this non-homology end joining, it is mainly used for knockout of the gene of interest. So if you want to knock out your expression of these target DNA, you can go for non-homology end joining, since it is error prone and it will bring about random mutations in the target DNA. So the protein which is synthesized after this non-homology end joining mechanism will be non-functional, or the protein will not be produced itself. It can be anything. It will be like this target DNA has been knocked off from the genome. Okay, that is non-homology end joining. Then next question was for the Cre recombinase. Okay. So Cre recombinase protein, it will catalyze the site-specific recombination between two DNA recognition sites. So this recombinase, that is a Cre recombinase, it is produced by the bacteriophage P1. Now the Cre recombinase, it will recognize a site called as the LOXP site. So when the gene is between the LOXP site, only then the Cre recombinase will function. Now depending on the position and orientation of this LOXP site, you can bring about inversion, deletion, translocation. Now here I have given deletion as example, but when you are discussing the part B questions, I will tell regarding insertion, deletion, translocation and all. So here just one statement they have given on Cre recombinase. In the same question paper they have asked for in part B regarding this Cre recombinase exact principle. Okay, that time I will tell you regarding inversion, deletion, translocation. Now here I will tell you regarding the deletion. Now if the LOXP site is in the same direction, that is this triangle is facing in this direction, this triangle is also facing in this direction. These are your LOXP site. This is the gene X that you want to delete. So the LOXP site are facing in the same direction. So the Cre recombinase, when you add it, what it will do is it will delete this gene X. So this is gene X deletion. Okay. Now when you are conducting the experiment in animals, that is this Cre recombinase LOXP experiment you are conducting in animal, one example I will tell you is how they conduct this experiment. One example I will tell you. They will transform a mice with the Cre enzyme, Cre gene, which will produce the Cre recombinase protein. So this is the promoter of the gene. This is the Cre coding sequence which will code for the Cre recombinase protein. Then another mice will be there which is having the target gene which has to be deleted, which is flanked by the LOXP site. Now they will breed these two mice, and this is the offspring of these transgenic mice. It will have both the genes. That is, for the Cre gene also will be there, and the one which has to be deleted flanked by LOXP site will also be there. So now this promoter, it will synthesize the Cre recombinase protein, and this Cre recombinase protein, it will act on the LOXP site, and this target gene, it will be deleted. Okay. So that is just one example how you do this, how you use the Cre recombinase technology for deleting the gene. Now come to the statement. So the following statements are made about transgenic mice technologies. So catalytically inactive Cas9, inactive. Okay, it is inactive Cas9. It doesn't carry out its catalytic action. That is, it will not bring about a double-stranded break. Okay. But it can form complex with the guide RNA. Okay. Fused to a transcription activator. Now transcription activator means it will enhance your gene expression. Transcription can be used to activate specific gene expression. Yes, 100% you can use. Now, how is it done? So, this inactive Cas9, it can bind to DNA as well as it can form complex with the guide RNA, and it is fused with the transcriptional activator. So transcriptional activator, I told you, it promotes gene expression or enhances gene expression. So as usual, you will design a guide RNA which is complementary to the target DNA sequence, and this target DNA sequence, it can be your promoter or an enhancer region of the gene, because you want to enhance the expression of a gene. Okay, for that you have to design a guide RNA sequence which is complementary to the promoter and or enhancer region. Now this guide RNA will direct this in Cas9, which is catalytically inactive but fused to transcription activator to the promoter region of the DNA, and when this transcription activator, Cas9, guide RNA, all these bind to the promoter region, because of the presence of this transcription activator, it will recruit other proteins to the promoter and it will enhance the transcription of that particular gene. So that can be made with the help of catalytically inactive Cas9 fused to transcription activator. So A is correct. Then using CRISPR-Cas9, a precise gene replacement in a target locus can be accomplished by non-homologous end joining repair. I told you after the Cas9 have cleaved the DNA, it can undergo either the homology directed repair or non-homologous end joining repair using the enzymes which are produced by the cell for these two repair pathways. So if you want precise gene replacement, perfect gene replacement, will non-homologous end joining repair work? No, because it is an error prone mechanism. Okay, so it can bring about deletion, duplication, insertion, etc. All these things can happen in non-homologous end joining repair. So for precise gene replacement, you need homology directed repair, which is error prone pathway. So B statement is wrong. That means the 3 gone, 4 also gone. Then using CRISPR-Cas9, many genes can be targeted at once by expressing many guide RNAs in the same cell. Of course, you can do, and that process is called as multiplexing. Okay, what is it called? It is called multiplexing. So this will, simultaneously it will target multiple genes or on a single genome. So what you do is multiple gRNAs which is having okay, I will show you. So this is your genome and you want to target gene A here, which is located here, a B gene which is located here, and you have got the C gene which is located somewhere else, and there are so many other genes. So what you do is, you know the sequence of this A, B and C. So you will synthesize guide RNA for A, you will synthesize guide RNA for B, you will synthesize guide RNA for C. Now all these guide RNA sequence you can insert into one single plasmid. Okay. Guide RNA sequences you can insert into one single vector. Then you will introduce or you will infect the cell with this plasmid. The gRNA will be synthesized, and Cas9 also would have been introduced into this particular cell. So the gRNA Cas9 complex will form, and this gRNA Cas9 it will target gene A. This gRNA and Cas9 it will target gene B. Then this gRNA and Cas9 it will target gene C. Like that, how many number of genes you want to target, you can use that by using this multiplex technology using CRISPR-Cas9. Okay, so in the same cell you can do this, you can use this multiplexing technology. So C is correct. Now next one, all chimeric mice derived by injecting engineered mouse embryonic stem cells into blastocyst will carry the transgene in the germ line. No, that is not correct. See, now you are having embryonic stem cells which is having your target gene or your gene of interest. You want to introduced into the blastocyst. So these are the embryonic stem cells with the gene of interest. Now these embryonic stem cells, they will be injected into the mouse blastocyst. Okay, now the blastocyst, these injected embryonic stem cells, they will integrate with the inner cell mass of the blastocyst. So this embryo which is having this embryonic stem cells injected into the blastocyst, that embryo you will implant into a surrogate mouse. Now this mouse, it will have cells from blastocyst as well as from injected embryonic stem cells. Okay, till so far you have understood. So now what they have told, a chimeric mouse derived by injecting engineered mouse embryonic stem cells into blastocyst. So from this surrogate mother, the mother she should give birth to chimeric mouse offsprings. Chimeric means they should have your gene of interest in them. Okay, so the surrogate mother, she should give birth to chimeric mouse offsprings. So these, what they are telling is, all chimeric mice derived by injecting engineered mouse embryonic stem cells into blastocyst. So these are the, when a mouse gives birth, it will not be just one offspring, some 2 or 3 or even more will be there, whatever. So all the offsprings will be chimeric. Okay, all the offsprings will be chimeric, but it is not necessary that whatever embryonic stem cells that you have introduced into the blastocyst will give rise to, will carry the transgene to the germ line of these chimeric mouse. It is not necessary. All the embryonic stem cells need not give rise to the germ line. Germ line means your sperm and the egg. That germ line, it need not necessary that these embryonic stem cells, whatever you have introduced into the surrogate mother, need not give rise to the germ line in chimeric mice. It need not always give rise to the transgenic germ line of the chimeric mice. Then next case is, these embryonic stem cells, they are carrying the transgene. Okay, these embryonic stem cells, whatever you are introducing into the blastocyst, if they are not carrying the transgene and they are introduced in and they go to the germline of the chimeric mice, still then you are not getting the target gene introduced into the chimeric mice. Okay, so all chimeric mice which are derived from these embryonic stem cells, they will not carry the transgene in the germline. Okay, so it is not necessary that these embryonic stem cells, whatever you have introduced into the blastocyst, will contribute to the germline in chimeric mouse. Some of the offsprings from that surrogate mother would have the transgene, but not all of them. Okay. So that is the so that makes option D wrong. Then in the Cre-Lox conditional system, the Cre recombinase is inserted into the gene to be knocked out. Cre recombinase is the enzyme that will bring about the double-stranded cut when the gene you want to knock out is being flanked on both the sides by the LOXP. Now, if you want to knock out one particular gene, and what you are doing is you are going to introduce Cre recombinase into that particular gene where you want to knock out, means it is already knocked out. When you introduce Cre, see, this is a gene that you are targeting. Okay. Now what is the statement telling? In the Cre-Lox conditional system, the Cre recombinase is inserted into the gene to be knocked out. Now you are in, this A gene will produce one specific protein. Now to this A gene you are introducing the Cre recombinase. So will the protein that is produced by A be functional? No. Okay, it will not be functional. So if you, so in this Cre-Lox system, the Cre recombinase will not be introduced into the gene to be knocked out. It will be I told you in the previous slide that the Cre recombinase is produced by one mouse, whereas the gene to be knocked out having the LOXP site, it is produced in an other mouse. Okay, only then Cre recombinase will delete this gene or it will be knocked out. So E is also not correct. So that makes A and C as the right answer. So 2 is the correct answer. Then next one, following statements are made regarding mapping population and principle of genetic mapping in crop species. You have to find out the incorrect statements. Okay, now first one. For self-pollinating species, both F2 and F1 derived double haploids can serve as mapping population. 100% correct. How are these double haploids produced? So double haploids, first you will give rise to the haploid plant. It can be through the breeding way or you can go for in vitro way. In vitro way, if I tell you, you will isolate the pollen or the ovules. Usually it is the pollen that they go for. So you will isolate that, grow it in the culture medium. This pollen is haploid, you all know it. So whatever plants that come out from this pollen regeneration, it will be homozygous. They will be haploids. Now they have to be converted to the double haploids. So for that what you will do is you will treat with the colchicine. So the chromosomes will be doubled. So this plant, it will be a completely homozygous line for all the traits and it will be diploid also. It is double haploid. Okay, so that can serve as a mapping population. All the F1 will be genetically identical and they can be used for creating mapping population. So A is correct. Then for cross-pollinating species, F1 population can be used for mapping studies. Of course, cross-pollination. So F1 can be pollinated to give rise to F2. F2 population can be used for linkage studies because they exhibit segregation of traits. So they can be used for mapping studies. So B is correct. Then bulk segregant analysis is often used to identify linked markers for complex traits with multiple genes and environmental influences? No. What is bulk segregant analysis? You usually cross in bulk segregant analysis. You use it for identifying DNA markers linked to a trait. So here if you are taking the condition of disease resistance. So plant A is with the disease resistance, B is with susceptible to that particular disease. So only very not complex traits, just differing in their phenotypic expression you are choosing. So you then you cross these two, you get the offsprings you phenotype, that is to see whether it is a disease resistant or is it susceptible, and you may get a range of phenotypes, that is it can show high extreme disease resistance, high extreme susceptibility, and intermediates also will be there. Now what you do is you will not take the intermediates into consideration. You will take the extreme ones. So one which is disease resistance, highly disease resistance, one which is highly susceptible. That two plants you will take. So group 1, group 2. Now you isolate DNA from all the plants which is highly disease resistance, pool it. That is the bulk DNA. That is bulk. So first bulk. Then group 2 plants, highly susceptible one. Isolate DNA, pool all the DNA from these plants, that is DNA bulk 2. Then you perform this high throughput sequencing, find genetic markers which are more, search for genetic markers which are more frequent in the bulk. So if bulk 1 is having, that is disease resistance is having the marker which is associated with the disease resistance gene, or else the marker is always linked to this particular disease resistance gene, you will say that this particular genetic marker, it is always associated with the disease resistance, whereas that case you will not see in the case of the susceptible one. So bulk segregant analysis, it is usually for simple traits, usually controlled by 1 or 2 genes, not multiple genes. If it was complex traits, like in the case of height of human beings, if you do for that, you will know that complex traits, they are controlled by many different genes, like quantitative inheritance, polygenic. So many different genes, and each gene it will contribute differently to that particular trait. One gene it will contribute more to height, another gene will contribute only intermediate amounts. So another gene will contribute high amounts for height of the individual. So it is for, so BSA will, you cannot use BSA for complex traits and also controlled by multiple genes. Now human height, you know, it is also influenced by environment. So environmental influences will also determine the phenotype or the height of the human being. If that is the case, then BSA cannot be used. It will not be used for complex trait with multiple genes, and these genes being influenced by environment also, it will complicate results in bulk segregant analysis. So bulk segregant analysis, you use it only for simple traits. And bulk segregant analysis, it relies, I told you, it is mainly seeing into the phenotypic differences between the two bulks whatever you have isolated. It doesn't even take into consideration the intermediates which you obtained after crossing. So C is wrong. Then in crop plant, sets of different breeding lines can be exploited for trait mapping. Yes. Different breeding lines means near isogenic lines, recombinant inbred lines. All these they can be used for identifying the marker associated with the trait. That is trait mapping. So D is correct. So A, B, D, okay, C gone. So wherever C comes, oh no, they have asked for the incorrect statement. So B, we have to see for uh, the incorrect statement. So C has to be correct. That is incorrect statement. So wherever C comes, it will be correct. Okay, so C is there in all the options. What about A and B? Okay, A and B are right statement. So wherever A and B comes, that statements will be wrong. Okay, D is also a right statement. So that makes 3 also wrong. So your answer naturally must be 4. Okay. Okay, what is E? Breeding methods such as single seed descent and pedigree selection promote genetic variability. 100% wrong. Single seed descent and pedigree selection, they are usually for increasing the homozygosity of a trait in your plant, and it is used for fixing that particular homozygous trait in the plant. So if you want genetic variability, then you have to go for the heterozygous condition, but you won't get heterozygosity in single seed descent and pedigree selection. They go for the homozygous one. So genetic variability will not be seen much in single seed descent and pedigree selection. So E is also a wrong statement. So your answer is C and E only. Then next one is your RT-PCR. Okay, real time PCR. Now real time PCR, it is used to quantify the target sequence and to compare the relative amount of target sequence between samples. So if you want to see, conduct RT-PCR for COVID infected and non-infected person, you can go for this RT-PCR. You can see the relative amount of the viral RNA in COVID infected person and lesser amount of viral RNA in the uninfected person. Okay, so it is a technique that and it also you can monitor the amplification of the target sequence at real time. You can see it on the instrument. Okay, or if it is connected to the computer via a software or something, there also you can visualize. So it will give the fluorescence based on the probe whatever you are using. Now even though you are using this fluorescent dyes and specific probes, there is a lot of noise over here. See this diagram, if you see, you cannot distinguish between the samples in this line. Okay, that is because of the noise it creates. So to bypass this background noise, what you do is you consider two values in RT-PCR. One is the threshold line. See here, this graph you see first. This is the threshold line and then is the CT value. Now before that I will tell you what is this baseline. So baseline, it is the PCR cycle at which the fluorescent signal accumulates. You cannot, it is not detected by the instrument. Okay, it's very highly complicated, too much noise over here. It is not detected by the instrument. Whereas the threshold line, it is the level of detection by the instrument. Okay, so it will distinguish the amplification. See here, you can distinguish once it crosses the threshold line, you can easily distinguish the samples, whereas here you cannot distinguish the samples. So that is threshold line. And before conducting this PCR, you can either you can set the threshold line, fluorescent value according to your wish, or else you can ask the software of the instrument to do it for you. So both manually and automatic you can do. Now see this amplification curve. This is one amplification curve. You will get many in your RT-PCR cycle. So this is the initiation phase, this is the exponential phase, and this is the plateau phase. So this is the initiation phase and it occurs during the initial PCR cycle. So these are 5, 10, 15, 20. These are the various PCR cycles. So at first PCR cycle you can see this initiation phase, and the fluorescence that is emitted at this initiation phase, you cannot distinguish it from the baseline. Here, since only one curve is there, you can see baseline here and the initiation phase. But see this diagram, the baseline and the initiation phase you cannot distinguish. So that is the initiation phase. Then you have got the exponential phase and the fluorescence will increase. See the fluorescent value over here, I have not written the values. It is increasing in the exponential phase, and then it will come to the plateau phase. Plateau phase is when all the reagents, whatever you have added for your RT-PCR, they have got exhausted and they have finished. So you won't observe any increase in the fluorescence, and it will form the plateau, and quantification is not possible in the plateau phase. Okay. So far you have understood. Now what is this CT value? So the CT value, it is the line, it is the value at which this amplification curve, it will intersect with the threshold line. See here, it is the PCR cycle at which this amplification curve, it will intersect with the threshold line. So here if you take, it will be around 11 or 12. Okay, the PCR cycle will be 11 or 12. So at that stage you will get the CT value, and you can detect a fluorescent signal when your sample reaches the CT value. So that is the real signal. Okay. So that is CT value, and one more thing I will tell you regarding CT value is the CT value, it is inversely proportional to the amount of target sequence in your sample. So if there is high amount of target sequence in your sample, then at lower cycles itself you will get the signal. Okay, so high target sequence, CT value will be less. It is inversely proportional. High amount of target sequence, CT value will be less. CT value corresponds to the number of cycles. Okay, so CT value will be less. But if the target sequence is less, then at higher cycle values only you will get CT value. So less target sequence, high CT value. See here in this graph, these lines and the green and the orange line, here high target sequence, here high amount is there. So at lower cycle itself you got CT value. See here for this also. But these two target sequence is less. So it is at higher PCR cycle you are getting the CT value. Fine. So that also you remember. Now coming to what is called as normalization. So normalization means, why you do is because these you get a lot of CT values. So somebody can argue that yes, these CT values, they are not because of your biological changes. Okay, suppose you want to see gene expression during different phases of plant growth. So one particular gene, it is expressed more in the fruit, whereas it is expressed less in the leaf. So if it more in the fruit means at lower PCR cycles you will get CT value, whereas in the leaf, since it is less expressed, you will get it at higher cycle number, CT value. So somebody is going to argue that your experiment is wrong. You cannot get always at higher values in the I mean, you cannot get your target sequence expressed in the fruit always just because it is showing lesser PCR cycle number. Somebody is arguing like that. So what you do, you use a standard. So standard is for normalization. Now this standard, it is also called as a reference. Okay, reference value. And this standard, whatever you are using, it will be your housekeeping genes. So they will always be expressing without much changes in the expression level. They are constant, they will show constant expression levels. So these housekeeping genes, they can be your actin or tubulin or ubiquitin or anything according to your choice. So for that you use the control, and all scientific experiments, they require a control. So here real-time PCR experiments also require a control. So to the person you are arguing, you can tell, see my standard values. The reference values, they are also showing the CT value at lower PCR cycles. So chances are there that my sample is not contaminated, or that my RNA extraction is perfect, and it is not because of RNA difference in extraction variation during the RNA expression that I am getting the CT value lower for the fruit and CT value higher for the leaf. It is not due to my extraction method or due to some technical variation. See the standard, it is showing CT value at lower PCR cycles. For the standard, you already know at what PCR cycle number it will show the CT value. So like that you can tell. So for that sake you will use normalization. Okay. Now come to the question. So equal number of particles of a mutant viral strain A. So there is a virus which is mutated, and the wild type strain. So wild type virus is also there. Were used to separately infect equal number of synchronized compatible host mammalian cells. So these two viral strains, the mutant and the wild type, they were infected to the mammalian cells. RNA from host mammalian cells were subjected to RT-PCR. So you extracted the RNA from the cells which were infected, and you have conducted the quantitative real-time PCR analysis. So what gene are you targeting? So first you are analyzing the viral gag gene. So for infected with the A as well as infected with the wild type, you have analyzed the CT values. See here, these are the PCR cycle at which this sample has reached its threshold, and this one for when this sample has reached the threshold value. So these are the CT values. Now you understood what is CT. And host beta tubulin after 24 hours. So you have used beta tubulin, which is a reference standard. This is for the, see the beta tubulin values for infected with A and infected with the wild type, there is not much change. Okay, the uninfected there will be 70. Beta tubulin doesn't vary. See, for infected with the A or WT or uninfected case, the CT value for beta tubulin, it is not changing, it is constant. So that is why you require a reference standard. Then how much of the gag transcripts in A infected cells change with respect to WT infected cells. So what is the fold change? That is what you have to calculate here. So they have given these values. We will see one by one. So for this you use the delta CT or the LIVAC method. So delta CT method, it is by first you will calculate delta CT, that is difference between the CT values of the sample as well as with that of the standard. Okay, then you calculate the delta CT, that is for CT of the mutant A and delta CT of the wild type. They you will subtract, then you will go for the fold change, that is given by 2 raised to minus delta CT. So this will give the relative expression level of gag in A relative to wild type. So first infected with A gag was having CT value 22, beta tubulin it was 17. Then infected with the wild type gag was 23, beta tubulin is 18. So delta CT for each sample. For A it is target minus reference, so 22 minus 17, so it is 5, and for WT it is 23 minus 18, so it is 5. Okay. Then delta CT you have to calculate. So delta CT A minus delta CT W. So 5 minus 5, that is 0. Now fold change will be 2 raised to minus 0. Delta CT. So anything raised to 0 is 1. So the relative expression level of gag A relative to wild type is 1. So A is equal to wild type. So there is no change. So the answer is no change. Okay. Then come to the next question. They have asked, they have shown the table of some cultivated crop and the chromosomal ploidy. This you have to know by heart. Okay. So banana, it is triploid. So A will be 2, A2, so 1 gone, and 3 gone. Okay, so A2. Then bread wheat, it is hexaploid, so B6, B4, sorry, B4 it is hexaploid, so 2 it is 4. Then brown mustard, it is tetraploid, so C3, that makes option 4 as the right answer, 2 gone. Then cauliflower, it is a diploid, and sugarcane, it is octaploid. So this ploidy of some cultivated crops you have to know. So A2, B4, C3, D1 and E5. Then next one is a problem. So a glucose biosensor exhibits Michaelis-Menten kinetics with a maximum current response of 250 microamperes. Michaelis constant Km is 5.2. If the glucose concentration is 3.4, what is the current response? So it is given, so the sensor's current response and the glucose concentration, it follows the Michaelis-Menten equation. So instead of V you are using I. Imax, it is the maximum current response. I is current response, Imax is maximum current response, S is the substrate concentration, and Km is Michaelis constant. So all the values they have given, substitute, and you will get 98.83 microamperes. So that is answer 1. Then given below are the names of techniques, that is column X, and column Y they have given the characteristic features measured for molecular characterization. So you have to match. So the first one is X-ray crystallography. Naturally it is diffraction. So everybody knows that. That means 2 is gone and 3 is also gone. So NMR chemical shift, that also everybody knows. Okay, that itself will give you the answer as 1. So if you know 2 of them, happily you can eliminate 2, 3 and 4. 1 is your answer. Then Raman spectroscopy, again it is wave number. CD spectroscopy, it is ellipticity, and colorimetry, it is absorbance, Beer-Lambert law. So even if you don't know B, C and D, only you know A and E, then also you can get the answer. Okay, see here, if it is A is 3 and E is 4 in case of option 4, but you know the answer as E is 6. So happily you can go for 1. So be careful when you are seeing the options also. Just by seeing that after X-ray crystallography, NMR, I don't know what it is. Why I will waste my time reading other options? No, see, some 1 or 2 you may be knowing. Based on that, if you want, you can go for the elimination technique and then you can find the answer. Okay, now next one. That is your type 1 and type 2 errors. They have given the null hypothesis. So choose the choice regarding the 2 types of errors made in statistical hypothesis. Okay. So what is this null hypothesis? Null hypothesis means it is a negative statement. Just remember that, negative. Everything is negative for null hypothesis. So what it states, if you are checking the you have isolated a new drug and you want to see whether it is having some effect on the blood pressure, your null hypothesis will be no effect at all, negative, everything negative. So null hypothesis also, drug is having no effective on the blood pressure. That is null hypothesis. Now there are 2 type of errors. One is the type 1 and the other is type 2 error. Now type 1 error, it happens when you reject the null hypothesis. Okay, when actually it is true. So null hypothesis, one example I will tell you, I am having the disease, but so what that is my hypothesis. So null hypothesis is I do not have the disease. I do not have the disease. So you do a medical test on me and you find that the medical test indicate that I am having the disease. So my null hypothesis is wrong. That is what you tell based on the medical test. But in reality, null hypothesis is correct. So what is my null hypothesis? I do not have the disease. So you reject the null hypothesis when it is true. So H0 is true. You reject null hypothesis. It is type 1 error. Okay. Now come to the type 2 error. Whereas here B option, if you see reject null hypothesis is correct decision. No. Here this null hypothesis is true and it is a type 2 error. So that is also not correct. Here null hypothesis is true and you go for the correct decision. That is also not correct. So C gone, B gone and D also gone. So if you know the first what is type 1 error, then itself you can come to the answer. Okay. Then next case. You fail to reject the null hypothesis when it is actually false. So null hypothesis is false. What is your null hypothesis? I am having the disease. So what you do? You do the medical test. It shows a negative result. You don't have the disease. But in reality, you are sick. Okay. So that is type 2 error. That is null hypothesis is false. Yes, you do not have the disease. That is what null hypothesis stated. So that statement is false. Just because your medical report showed that you are not having the disease, you thought null hypothesis is correct. So in type 2 error, you fail to reject the null hypothesis, or else you accept the null hypothesis when in reality it is not correct. It is false. So it will give rise to type 2 error, also called beta error. So A is the right answer. So option 2. Then okay, next one is your PCR. Now in PCR, this is the full length genomic DNA. This is red one, that is the target sequence. Now when you conduct PCR, you add all the reagents, set the PCR to its denaturation condition, then annealing, then final extension, and then final extension. So first denaturation, these two strands separate, then annealing, primers bind, then extension, polymerases are going to start synthesizing the DNA, then final extension, polymerase will stop extending, finished. So you set the time for each of these. So denaturation maybe 1 minute, then annealing maybe another 1 minute or even less than that seconds. Then extension, final extension, all these will be 1 minute. So the DNA, whatever you are providing, it is genomic DNA, or suppose I am taking that as an example. You add all these reagents, then you start the experiment. Once it reaches final extension, the polymerase, it has a tendency to keep on going on. Okay, it will go on. But at the time the final extension stops, that is at 1 minute, if you are set the PCR at that condition, the polymerase will stop extending. The it has not completely extended the genomic DNA. Okay, you have set the time for 1 minute. You know your target DNA sequence is only of this much, and you know that it requires only 1 minute for the polymerase to synthesize that length. But the polymerase will keep on moving all along the length of the target DNA. But it would not have reached the end of the target genomic DNA, whatever you have supplied. So it will reach up till this green portion, and these are the extended. They may not be of equal length. So these are the extended flanks that you get. The other portion you will not get. This is the next substrate for the next cycle. So here some portion of your genomic DNA other than the target sequence will be present. Okay. So again the PCR is continuing. Again by final extension, it will stop. It has reached here, but some portion of the green region also is being incorporated along with the target DNA sequence. That is up to your third cycle. No, first, second, yes, till third cycle. For the third cycle, this will act as the substrate. Now again you do the PCR cycle, but this time there is not much DNA to be extended. So up till the target DNA sequence, only the polymerase can extend till the end of target DNA sequence, not beyond. There is nothing much left out to go beyond and extend. So as the cycle number increases, you will get a fully pure target DNA sequence. Okay. So that's it. So first, second and third. By third cycle you may get third or fourth depending. So you will get a fully amplified target DNA sequence. These regions which are not required by you, they will not be amplified. So these whatever you got, these intermediates, they are not correct. Okay, because it has got some portion of the other genomic DNA also, but they will be present in lesser amount. So when you do the agarose gel electrophoresis after you have set some amount of cycle for the PCR, you will get a thick band with the target DNA sequence. Okay, band intensity will be more than. These bands, even though they may be present, they may not be that much visible. They may be very light, that is why they are not visible with the agarose gel, and also depending on the concentration of agarose, whatever you are using, they are not much visible. But if you stop after 10 cycles and then you run the gel, maybe you can see these bands. Okay, so that is not our question. Our question is regarding the formula that you are going to use to calculate the target DNA sequence. Okay, so this is the question. A polymerase chain reaction with all necessary reagents was carried out to amplify. This is the 200 base pair target region. This is your double-stranded DNA of the human Y chromosome. So this is the Y chromosome DNA. It is comparatively very long, and your target region is only 200 base pair. So how much PCR was started initially? You have used only 100 copies of the double-stranded DNA as a template. So this is the double-stranded DNA template. This is the target region. The primers are annealing there, and you got, and this is 200 base pair. So this is the 200 base pair fragment that is produced at the end of PCR cycle. So assuming 100% PCR efficiency. So PCR is 100% efficient. Tag polymerase 100% perfect. Annealing temperature 100% you have set. There is no primer dimer formation, etc. Perfect. Okay. So 100% PCR efficiency. How many copies of the desired 200 base pair fragment consisting only, that is the keyword, only the target region. That is only this region. This portion shouldn't be there. These portions shouldn't be there. That is your intermediates shouldn't be taken into consideration. I told you already how that intermediates are coming. Okay, so these are the intermediate. They shouldn't be there. Only this target sequence should be there. 100% it should be target sequence only. Would be produced at the end of third PCR cycle. So after, I told you, after 3 PCR cycles, the DNA will be mainly the target DNA which is going to be amplified. These portions of the DNA will not be amplified as happened in first and second. That is your intermediates will not be amplified. So you have a formula for that. That is number of target regions after n cycles. Small n, remember that. So it will be capital N into 2 raised to n minus 2n. So capital N is the initial amount of target DNA template. Here it is 100. 2 raised to n means small n. Okay. 2 raised to small n is your number of cycles. Okay. So 2 raised to n means the total DNA copies. That is, you will have your template DNA, that is this whole DNA sequence. Some amount will be there. So template will be there. Intermediates I told you, which will have the target DNA sequence flanked by a small piece of your double-stranded DNA, that will be there. That is your intermediate, and as well as the desired product. That is your 200 base pair fragment, that also will be there. Minus 2n. So this 2n, it represent the copies of template and intermediate fragment, but it doesn't contain the desired product. So 2 raised to n, it is having template, intermediate fragment and desired product, whereas 2n, it is having only the template and the intermediate fragment. So subtract these two, you will get the desired product. So into the initial amount of DNA, you will get your number produced at the end of third PCR cycle. Only target DNA, okay, fragment. So that will be 200. That will be the number of copies of the desired fragment produced at the end of third cycle. Now if it is fourth cycle, 2 raised to n, you put it as 4. So 2 raised to 4 minus 2 into 4. You will multiply by the initial number of copies that you have added. So that will give you the number of target regions after n cycles. So that's it. Thank you for watching my channel. If you want previous year's question papers of CSIR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, see for CSIR, and you will find them there. So thank you for watching. Please do like, share and subscribe. Thank you.